morning, everyone. I'm Lori Burrows from the IIDR, and this is our February 2022 edition of IIDR joint rounds. So I'm very happy today to have Dr. Des Pernica and Dirk Hackenberger speaking. So I'm just going to give you their bios quickly. Dr. Pernica is an associate professor in McMaster's Department of Pediatrics, where he also serves as the head of the infectious disease division. His research interests relate to the optimization of the diagnosis and treatment of pediatric respiratory infections, enteric infections, and sepsis. And today he'll be talking to us about diagnosing sepsis in resource-limited settings. Our second speaker today is Dirk Hackenberger. He's a PhD candidate in the Wright and Poinar Labs at the IIDR. He is working to create novel techniques inspired by ancient DNA analysis to use for the sequencing-based diagnostics and characterization of infectious diseases. Today, he'll be speaking to us about using DNA sequencing as a sepsis diagnostic. So I will turn the floor over to Des, who is our first speaker. Thank you both for doing this.
Thanks so much for having us. So, we're talking about sepsis today, and for those of you who don't know, sepsis is basically just getting sick all over because of an infection. I mean, I think we've all seen the medical show part where the person comes in and everybody rushes around and does things. As I don't need to say it for this particular audience, but obviously there are numerous causes of sepsis, many of which are bacterial, some are not. And if you die in a hospital, there's a good chance that it is related to sepsis. Now, this is not just this is not just a Canada problem, but a problem in most of the world, right? And what we have learned over decades is that the way to treat sepsis is to administer fluids and antibiotics as quickly as you can. This really came to the fore of publication of this paper 15 years ago, where they found a striking linear relationship between the time to antibiotics and mortality. Mortality is the one outcome that everybody cares about, right? Patients care about it, physicians care about it, healthcare policymakers realize that it's important. And even though there were problems with this particular paper, right, this association has been verified in subsequent very large studies, like this multi-center one done in New York State, where they found an association between the rapidity of care, okay, and mortality. Interestingly, there was no relation with how quickly the fluids went in, but it was shown that time to antibiotics drove most of this risk. And it has been verified in other settings as well. This is another recent study looking at the time between when people got to the emerge and got their antibiotics, and found again a very close association with mortality and time to antibiotics, whether that mortality was 1 year mortality, in hospital mortality, 30-day mortality, 90-day mortality, right? So we all agree mortality is bad. Antibiotics are good, obviously. And again, for this audience, I don't need to talk about this. They have to be active against the infecting pathogen to provide any benefit. And so the question then is how do you make sure that the antibiotics that you're choosing are the right ones? Now, most of this answer is bacterial diagnostics. And so let's just talk about what will happen from a clinical standpoint if at the completion of this talk, I become sick and go to the Juravinsky emergency department, for example. I get there, I have to get sick enough to make it think that it's worth my while to go to the hospital. Then I go to the hospital, then I go into the emerge and I see the triage person, and then I wait, and then I finally see a physician who decides that I look sick enough that a blood culture is required. And then I take 40 mils of my blood, divided equally among 4 of these bottles. And then they get sent to the core lab and they wait there for a little bit. And then they get driven to the general, and then they wait there for a little bit. And then they get accessioned and put into an incubator, right? Where the bottles get gently shaken until such time as that there is sufficient bacterial growth, say, 8-9 hours, that the bottle becomes positive, rings off, a tech comes and gets the bottle out of this, takes a few drops off, stains it with pretty colors in this technique that's like 130 years old to get a gram stain result, which is then, called to the place where I am in the hospital so that doctors, can potentially make antibiotic changes based on this. But we still don't have identification or susceptibility, right? So it needs to be plated, and it takes some hours before there's a tiny speck of colony enough to shoot with lasers. And often the mass spec can give you an identification, right? But you don't get a susceptibility until you take a little bit of the colony, put it into one of these automated machines, wait for another, 8 to 16 hours, and finally get the full shebang, right? Confirmation of identification and susceptibility. And so then you can be sure, right? And so just to look at it in a graphical way, these numbers are like the number of days since I developed sepsis. And you can see for the first day or two, we're just working on what the physician thinks is the best choice. We really don't have definitive ID and susceptibility for a day or two, right? And so in the time frame of this part of my medical journey, right, they are not, no diagnostic is helping these people. And this is why, selection of antibiotics empirically is a thing. Now, I should highlight that, it makes it sound like I'm saying, oh, this stuff takes too long. I think that our lab really works quite well. We have amazing help in the form of the technologists, the medical microbiologists, et cetera. In almost all of the world, things take much longer than they do in Canada. And so if you look at, for example, Botswana, right, you are not getting definitive IDs for 4, 5, 6, 7 days, right? And so the system that we have is pretty finely tuned. Having said that, again, we need to talk about, like, empiric antibiotic selection is a thing. And since people die if they don't get the appropriate antibiotics quickly, you think that you need to pretty much start broad. You need to start broad enough that you're likely to get, that the antibiotic that you pick is likely to be effective against the pathogen causing the infection. And later, when you know what it is, you can then narrow the spectrum. Now, the problem is, how broad is broad enough? And so some people would say, well, you should just jam as much as you can in there in an effort to save this person's life. The difficulty is that there is potentially too broad spectrum, and some people don't think about this because really lives do matter. The issue is that empiric therapy should be narrow enough to minimize not just cost and the likelihood of adverse drug reactions, but what we have seen consistently all over the world is that the unfettered use of broad spectrum antimicrobials will lead to increases in circulating antimicrobial resistance, which just makes the problem worse later on. And so I think, again, I know I don't need to convince anybody here of this, right? That overly broad empiric therapy might appear safe, but is probably more dangerous. And so when we are talking about prescribing in the context of sepsis, we need to get active, effective, reasonably broad spectrum antibiotics started quick, and then change to sort of optimal, sort of as narrow as possible antibiotics as soon as we have the information with which to be able to do that. And again, what happens at a clinical level is that doctors sort of try and use their diagnostic abilities to figure out what kind of infection syndrome the person has, pneumonia, cellulitis, whatever, and then infer what pathogens are most likely to be causing that, and then use local knowledge about circulating resistance within those pathogens to pick the best empiric strategy. Okay, now the rapidity with which diagnostics are used will then enable a shift to effective antibiotics if the empiric antibiotics didn't cover the organism at all. And even if it did, diagnostics will enable narrowing to optimal antibiotics. Now, again, in our context, we have all these things, right? that work relatively quickly. We can put together the immense resources of the diagnostic laboratory plus the information that we have in terms of local epidemiology and be able to craft empiric strategy relatively well. In other countries, it is nowhere near that easy. And so let's talk about Botswana a little bit. This is obviously a map of Southern Africa. Here is where Botswana is. And as a country, it is a middle-income country. So it doesn't have no money. You can see here's a GDP list. On this part of the list is also like, Cuba, Bulgaria. So it's much better, okay, than the poorest countries of the world, but nowhere near Canada, Belgium, New Zealand, for example, right? This is the University of Botswana. It looks very fancy. All this stuff was created in the last 15 years. This is a regional hospital, still looks fancy, right? The main referral hospital is somewhat older, and so, it looks more like this kind of thing. The issue with all of these healthcare institutions in Botswana is that it's sometimes easier to make nice buildings than to have things inside, right? And so what you're dealing with often is wards with little to no infection control, and things run out all the time, whether it's saline or oxygen or antibiotics or whatever. And the same holds true for the microbiology laboratory. Anybody who's been to the Hamilton General would be amazed at the stuff that there is there for clinical microbiology. Like there are numerous automated, we've talked about this, identification systems. Mass spectrometry has really changed things since I was a fellow, right? And you have legions of highly skilled technologists. Not only does Botswana have many fewer people, because skilled workers of any stripe are harder to keep in low resource environments, but there is no automation here, all right? And so they do things the way that they did, 80 years ago, biochemical reactions. Now, the issue with that is that you need lots of consumables, and supply chains are always a problem. This is pre-COVID, supply chains was a problem. Stuff is always running out, which makes clinical bacteriology very, very difficult in this environment. And again, this is what it looks like in Canada. It is much, much worse in Botswana, where you don't have this automated stuff and you're always running out of this disc or this reagent or this thing. And so you ask yourself, we have a finely tuned machine here in Canada, but in Botswana, if you had something that was simple and self-contained and didn't require 17 different liquids, would it improve care? Would a simple rapid assay enable physicians to get answers sooner and save lives? And there are simple assays, right? So this is one of them. There's tons, right? This is just one example. This is the BioFire film array, where, it's a little machine the shape of a bread box. It fits in any lab, really, and it just has one consumable, these cartridges. And they are so simple, okay, that my kids could probably do them with very little training required. Now, it doesn't test for everything by no means. It tests for a lot of things that cause sepsis, right, in terms of bacterial pathogens. So you can think, well, maybe we should give it a whirl. Other people might say, if you have these problems, why do you need to do a clinical trial? Obviously, it will be helpful. The issue, okay, is that even though this is simple and easy and whatnot, these little packets are expensive. So these are $100 a shot. We would never be able to afford this in Canada, because with economies of scale, with all the skilled personnel that we have in the upfront capital, things, it's cheaper to do things by mass spec, right? But Botswana is a different story, and a lot of environments across the world do not have the kind of infrastructure, both human resource wise and capital wise, that we have to be able to make our laboratory run. The other questions that we had was, if we are able to test stuff faster, will the physicians at this center at Princess Marina Hospital in Gaborone in Botswana actually prescribe better antibiotics sooner? And if they do, will it actually result in PMH patients doing better in this context? Okay, because if it isn't, the government will not pay for these things. And so this was our study question for our randomized trial for patients hospitalized at Princess Marina Hospital with a positive blood culture. Will the use of a molecular multiplex panel in addition to standard care decrease the time to optimal antimicrobial therapy. So definitive, so best possible therapy. Basically what we did is anybody who had a positive blood culture within the study period got randomized as soon as we made sure that they were still alive, right? And they hadn't been previously enrolled. They were randomized to either the new test plus the standard workup to the standard workup alone. For those who got randomized to the new test, what we would do is we would run it, usually get it that day. We would then take a piece of paper with the organism that we identified plus antibiotic recommendations and do the best we could to find anybody from the team to tell them that their patient had this result and give them antibiotic recommendations. The standard, okay, is that gram stain results get called to the ward, and then clinicians have to check the computer for the definitive identification of susceptibility that comes days later. The outcomes that we had, their primary outcome was time to the optimal antimicrobial therapy. We also wanted to find out how soon patients got started on any antimicrobial therapy that worked, right? The effective antimicrobial therapy and time to identification of the pathogen. What we found was we unfortunately were underpowered, okay? We had fewer patients than we thought, and everything stopped at COVID. So we were underpowered to begin with. Just to give you an idea of who these 311 people were, they were very sick. So a fifth of the population, study population, died in the hospital. And further compounding our power problem, more than half of these blood cultures were contaminated, right? And so you can't adjudicate an outcome if you have coagulase negative staph in the blood culture and it's not the actual problem. Now, these are the results, okay? These are survival analysis curves. So what you can see here is on the y-axis is the proportion of participants in each arm that reached the outcome. And this first thing is time to pathogen ID. So what you can see here is the blue line is the rapid test arm, the red line is the standard arm, and the rapid test did much better at identifying pathogens sooner. So you got a 2-day head start in pathogen ID with the rapid test. Unfortunately, when you look at the time it took to get people on effective therapy, these lines pretty much overlapped. So even though clinicians got these results faster, the patients didn't get started on effective antibiotics any sooner. And this was our primary outcome, time to best possible antibiotic therapy. And this is a total overlap, right? Somewhat more depressing, we followed people for 14 days. By the end of the 14-day study period, almost 70% of participants in both arms, okay, were not on the best possible antibiotic therapy. So what did we learn from this whole study? We have a test that's validated, that works fast, that's easy to do, but it was not enough in this particular context. And some of the reasons were the following. A majority of the participants were in the NICU, the neonatal intensive care, and there they have a real Klebsiella pneumonia ESBL problem, for which the only effective therapy accessible is meropenem. The optimal therapy accessible is meropenem, and so everybody gets started on meropenem. If infection patterns are really this homogeneous, it doesn't matter if you have a new, better test, right? Because meropenem is probably the answer in that context. Outside of the NICU, or where this isn't the problem, the other issue that we ran into, okay, is that even if we found the clinical team and brought them this piece of paper that said, this is the thing causing the problem, we suggest you change it to this, they would often say, thank you, because we were in Botswana, and then maybe not do anything. Because if clinicians are not used to having actionable results from the microbiology laboratory, if they're just used to doing what they're doing because that's what they do, then it takes a long time to really get people in the mode of changing their therapy based on lab results. And then, even if we did find people who were interested in making a change, right? If they were not empowered to do so, if they were worried about getting in trouble, often they wouldn't change things. And finally, even if somebody was willing and able to make a change, if the hospital was out of that antibiotic, it doesn't matter that we had the test. Okay, and so this was eye-opening for us, for this platform, for this context. The details of our study are obviously specific to Botswana, but the issues are the same, right? And the thing is that sepsis is really important And I think diagnostics have a real possibility of improving the way we deliver care and improving the way we prescribe antibiotics. But the clinical ecosystem is critical in terms of how much benefit there will be. We need to work with all aspects of the healthcare system, the physicians, the supply, the people who govern supply, the pharmacists, to really put the conditions in place so that we can make improved prescribing feasible and actionable for as many patients as possible in an effort to save lives. And that's what I got. Did I run over? Am I still good?
Perfect. Thank you. All right. So hi, everybody. Thanks for coming. Thanks, Dr. Pernica, for the fantastic talk, as always. Today, I'm going to be talking about leveraging DNA sequencing as a sepsis diagnostic. My name is Dirk Hackenberger. I'm a PhD candidate. I'm co-supervised by Dr. Wright Poinar and Dr. Jeffrey Wright. I think the link between Dr. Jeffrey Wright's research, who works primarily on antibiotic discovery and antimicrobial resistance characterization, the jump from that to sepsis diagnosis is not a huge one, though it is a bit of a jump. The link between Dr. Wright Poinar, who works primarily with ancient DNA, is not quite as clear, so I wanted to make that a bit more explicit before moving on to the deeper aspects of the presentation. Ancient DNA and pathogen DNA in plasma specifically are actually, though they seem only orthogonally related in that they're both DNA, there's actually a plethora of parallels that exist between the two. Ancient DNA is extremely short in damage due to long-term breakdown. As you can see down here, this is a mammoth skull that was found in Michigan. And you can imagine that this environment isn't exactly a -80 freezer for DNA preservation. The DNA breaks down due to hydrolytic and oxidative mechanisms, and it's just, by the time we get our hands on it, it's not in good shape. On the other hand, pathogen DNA in plasma is extremely short in damage due not to long-term breakdown, but rather to short interactions with the immune system. For example, neutrophils phagocytosing a MRSA bacterium here, they bombard them with reactive oxygen species and other damaging molecules that result in, again, extremely short and damaged DNA molecules. Additionally, ancient DNA is present at low levels against microbial background. You can imagine in here, this mammoth skull with its very ancient and degraded DNA is going to be drowned out by all the members of the microbial community that surround it in the dirt and everything else. The pathogen DNA plasma is also present at low levels against a human background. As you sequence DNA from a human sample, you're just going to have mostly human DNA, and it's going to be difficult to pull out the actual bacterial or infecting organisms DNA for diagnosis. And this is something I'll talk a little bit more on a bit later. So based on these similarities, what we want to do is exploit ancient DNA techniques that have been used and developed specifically for ancient DNA and apply them to pathogen DNA in plasma. Just to show this isn't too harebrained an idea, I want to show this paper, which is an example of using ancient DNA techniques to sequence pathogen DNA in blood, specifically single-stranded library preparation, a technique that was initially developed for sequencing and processing ancient DNA, was used to better detect microbe-associated cell-free DNA in transplant patient plasma. You can see here the frequency histogram for the different read sizes in these samples that they sequence. The difference between the single-stranded DNA library prep and the double-stranded DNA library prep is incredible. There's something 70-fold increase in the total number of reads in this blue line, which is single-stranded, red line, which is double-stranded. And moving on to the culture-based diagnosis of sepsis and what we're trying to do, as Dr. Pernica was saying, culture-based diagnosis is often slow. When we're looking at the standard microbiological testing timeline after sample acquisition in the lab, we're looking at sometimes in excess of 2 days. The other issues with it is that it has low sensitivity and it can't detect certain organisms such as viruses, unculturable microbes, etc. On top of that, AMR profiling takes extra steps. Our goal for my project during my PhD is to develop a robust and sensitive diagnostic for sepsis that delivers identification and AMR results simultaneously in less than 8 hours after sample acquisition. So just to give a little DNA sequencing diagnosis primer before we move on to the bulk of this presentation. A DNA sequencer generates something that are called reads, and these are relatively small pieces of DNA, usually less than 500 base pairs. The DNA in a sample will be representative of the organisms therein. And though this seems like kind of an obvious thing to say, what it's really trying to say is when you're sequencing anything from a human sample, whether that's whole blood, blood plasma, cerebrospinal fluid, anything, the vast majority of the reads are going to be background reads that you can't do anything about. Or rather that you just don't want to have in the sequencing data because it's not informative to your target. The target up here is actually a vast exaggeration. Typically in low concentration samples, we're going to be looking at a proportion of target associated reads, often less than 0.0001%. So these are some of the problems that exist with DNA sequencing. Before, just to show the power of it though, I wanted to use this paper as an example. This paper is from a group in Germany in 2019, where they're looking at the enhanced performance of next generation sequencing diagnostics compared with standard of care microbiological diagnostics in patients suffering from septic shock. So sequencing versus standard culture practices. And the main findings from this paper were that they took 256 blood plasma samples from 48 patients with sepsis at different time points. They attempted diagnosis with blood culture and sequencing, and at sepsis onset, blood culture had a positivity rate of 33%, whereas sequencing had a positivity rate of 71%. Determined by an outside panel of experts, 96% of positive cases were plausible, and 53% would have led to a change to a more appropriate therapy. And so seeing these findings, and of course there's others in the paper that I'm not brushing on here, but seeing these findings specifically, one could be forgiven for thinking, why are we not using DNA sequencing more often? And there's a few simple answers to that. And number one is speed. In the paper that I just showed you, they sequenced using an Illumina machine. This is the gold standard sequencing technology. It's used all over the world, and pictured here is a HiSeq 2500, the specific model of machine they use to sequence the data in the paper. The downside of this is that it has a runtime of around 60 hours. As Dr. Pernica was saying, this is an unacceptable amount of time to be able to diagnose sepsis. So ideally, we want our results to be as short as possible, realistically less than 8 hours. So the upside of this machine, however, is that it generates enormous amounts of data. So with this machine, you can generate 25 to 30 million reads per sample. On a single run, you can generate up to 300 million reads. So that's the benefit and the downside of this machine. The way that we can fix this problem of speed, though, is to simply use a different machine. Ion torrent sequencers, pictured here by Thermo Fisher, produce results in less than 3 hours. Fortuitously enough, Dr. Michael Surratt and the Pharmaco Metagenomics facility have recently acquired one, and it's something we're hoping to use in future experiments. However, the downside of these is they can only generate 2 to 3 million reads per sample. So this leads to another problem. When you're decreasing the total number of reads that you're going to be getting out of a sample, so a 10-fold decrease from Illumina to Ion torrent, you're going to be decreasing the total number of reads that are aligning to your target from an already small proportion. This means that you're going to be decreasing the sensitivity, which is the second problem of the protocol, because with a fewer number of reads aligning to the target, you're going to have less statistical power that's able to distinguish that from, say, a true positive from something like a false positive due to contamination or just random statistical noise of reads aligning to other organisms. So the question becomes, how do we increase the number of target reads that we get in the sample? And the answer to this is actually fairly simple, and it's a protocol that we use often in ancient DNA analysis. And it's something called hybridization-based enrichment. So to increase the number of reads in a sample, all we have to do is bias the library more towards that target organism. So we get a total number of reads that are greater for that target organism relative to the background DNA. So this protocol employs something called biotinylated RNA baits. For the purposes of this presentation, you can think of them as specific fishhooks that pull out specific target molecules. So we can see here we have blue for blue, red for red, purple for purple. And then all this black DNA here is the background non-target DNA. In this case, it would be human reads. What these fishhooks can do is that they can bind specific target molecules and preferentially retain them relative to the background, increasing their proportion in the sample. So what this looks like is that you let these fishhooks find their DNA partners in solution, they bind these targets, and then based on the biotin tag, we're able to actually grab those fishhooks and preferentially retain them throughout different wash steps while the background DNA is largely removed. During subsequent amplification steps, we result in a final library that has a far higher proportion of target molecules relative to non-target molecules. So we have a far higher proportion of what we want to sequence versus what we don't want to sequence. There are some limitations to this protocol, unfortunately. Typically, this technique is used to enrich DNA from individual species against a background. In an ancient DNA context, we might be trying to enrich for a mammoth mitochondrial genome against the vast microbial background that I was talking about earlier. However, the issue with sepsis is that it can be caused by a multitude of species. This figure here is something that I generated a little while ago, and it's just looking at the proportion of causative genera in different culture-positive patients with sepsis from two different data sets. One thing I want to point out beforehand, the white writing doesn't mean anything special. It's just easier to distinguish this from the dark background, and then the black writing is just easier to distinguish against a light background. For the first paper here, McGill et al. In 2014, they were looking at the proportions of causative organisms in sepsis, specifically in hospital-acquired infections in a cohort of patients across the United States. Vincent et al. In 2009 were looking at all infections in hospital, not necessarily just those that have been acquired in hospital, and this was a cohort of patients that came from across the world. And looking at these, we can see just from the causative genera, there's not a huge amount of genera that cause most of sepsis cases. However, there are 10 or so, and this doesn't actually account for the individual species themselves. The other issue with this is that we can see in this other organisms group in both of these charts, there is a very large piece of this, though not the majority, that are caused by these other organisms. And this is anything that's less than 2% of total cases. So even if we were to design a set of baits that are able to enrich for these top 10 causative genera, these other organisms are still going to be missed out by that bait set. And this means that we're going to be missing out on whatever this may be, 15 to 20% of total cases, which is not something we want to do. This doesn't even factor in the culture negative tests or the culture negative patients, or the unculturable microbes, viruses, etc., that also can accompany sepsis. So what we need for this is we need a way to design bait sets that are able to enrich not only for the individual species from what we know to cause sepsis, but all these other organisms here and the unculturable microbes and other things that we don't know cause sepsis yet at this point. And luckily, bioinformatics extraordinaire from our lab, Zachary Dixon, was able to do this just recently, which resulted in a paper published in Cell Reports Methods late last year. What he was able to do was reinvent bait design to target identifying regions of pathogen-associated DNA across genera and species. What this means is that he was able to design baits that were able to enrich specifically for unique identifying regions of individual species, as well as conserved regions that are targeted across genera. By doing so, we can enrich for known species by targeting these unique regions for enrichment, as well as unknown species from the known genera by targeting conserved regions. So this way, we're still getting everything that we know should be there. However, we're still able to pick up on things that we aren't 100% sure or that we aren't expecting to be there, so long as they're they have some sort of conserved piece of DNA from that genera. This is a common limitation with enrichment as well. Usually you can only sequence what you know is there. This way, we're still able to at least detect what we don't know would be there. Our newest set of 19,000 unique baits can enrich and identify 1,824 different genomes across 78 species and 33 genera. And these were actually just finished design on Friday, so this is a fairly recent development. When we looked at the verification of the original set of baits that Zach designed for enriching for sepsis, we used the newly designed bait set to enrich for bacteria spiked at different concentrations against blood and sequence the resulting samples. So we have 4 samples here: the blank, low, medium, and high. The blank is blood only, low is 10 CFU per mil of these bacterial species spiked against blood. medium is 1,000 CFU per mil, again, of these bacterial species against blood, and then high is 1,000,000 CFU per mil. SG here on the left for each of these bars means shotgun, so no enrichment. This is just standard sequencing. And then ENR means enriched, so that's here on the right. And this is looking at the relative proportions of each of the target aligned reads for these different bacteria in these samples. One thing I do want to draw your attention to is just the different y-axes on these different bars. This is simply a result of the vastly different input that we have in each of these samples. So that's all there is to that. What we can see here, though, is that we have very considerable enrichment in each of these bacterial species going from the shotgun to the enriched data. So enrichment is definitely working. On top of this, we're able to distinguish, if we look at the starting quantity of shotgun DNA here in the low sample relative to the blank, we actually have around a 10-fold higher amount of starting DNA in this shotgun data relative to the blank sample for the low with the 10 CFU per mils. This means that we can distinguish these low concentration samples that I was talking about at the beginning of the presentation from a blank. As I was saying before, we need more reads to align to the target organism to successfully do so. What we're seeing here is that we're seeing this signal in the blank sample, even though theoretically there should be none. This is just from potential small amounts of contamination or any one of a number of things. It could be statistical noise in the data. But this is something that's very, very difficult to get rid of. So we need to be able to distinguish a true positive from something that looks like this, which we can do. So the story so far, Illumina sequencing takes far too long to be able to diagnose sepsis. Ion-Tor machine lets us sequence faster, however, with a lower throughput, which limits our reads. Enrichment allows us to maintain statistical power with a lower number of reads by biasing the sample towards the target. So we have a starting sample that looks like this, an ending sample that looks like this. So we're still able to get the same or more reads aligning to our target organism with a smaller amount of total reads that we're getting from the sample. And so, as you can imagine, and as has happened so far in every other step of this protocol, the introduction of enrichment has led to another problem, and it's one that we've seen before. It's speed, again. The enrichment protocol typically takes more than 24 hours. Most of this is in the hybridization step, which typically on its own takes around 16 to 24 hours. This is the step in which the baits, so these specific fish hooks, find and bind their complementary partners. So the time it takes for the blue to bind blue, red to bind red, purple to bind purple. And luckily, there is actually a fix for this. And in this case, alcohol could in fact be the solution. Not to drown our sorrows in about the complications and constant problems of this project, but rather to speed the hybridization step. A group in 2010 found that adding ethanol to an aqueous hybridization buffer improved speed of enrichment, or speed of hybridization, 70-fold. They did this by using something called beacon probes. Beacon probes are these long loop structures connected by a hairpin at the ends, so that when they're not bound to their DNA partners, they adopt this conformation in which a quencher is close to a fluorophore, which results in any inhibition of any fluorescent signal coming from this. When it binds its DNA partner, it separates the quencher and the fluorophore, which results in a trackable fluorescent signal. So you can track hybridization as it occurs. And what this group found was that in 56% ethanol, relative to in water, there was a 70-fold increase in the speed of hybridization relative to the water, so relative to no alcohol at all. This could potentially be hugely powerful, because if we're able to capture this 70-fold increase in the speed of hybridization, it would mean that we can achieve a 24-hour hybridization in just 20 minutes. So this could be extremely powerful. On top of this, the alcohol, or the ethanol specifically, didn't affect the final equilibrium of the bound versus unbound products. And on top of that, it even increased the specificity of binding. So based on this, ethanol seems to really be acting as a catalyst in this situation, speeding the interaction of hybridization rather than some sort of modifying the reaction in some other way. So this is now getting into our current work, and this is unpublished and unpublicized. So right now we're testing a 1-hour hybridization in the presence of ethanol using qPCR. A caveat here is that this is completely preliminary work. I was hoping to have sequencing data ready for this presentation. However, it is only going on the sequencer as we speak. So unfortunately, things don't always go as planned in the lab. So the PCR results we have now are good as, I think, as a preliminary model, however, not fully indicative of the final results. So it could be better than the results we have here. It could be worse, but this is just something that I want to make you aware of beforehand. So looking at the sample that we have here, what we're doing is that we're testing and quantifying the background and the target retained during enrichment before and after this hybridization step. What we're doing is we have a sample here, which in reality is a ancient mastodon sample. It's not important to the presentation itself, but it's still, people sometimes find that kind of neat. And essentially what we have is the background reads here, which in red, which are, they shouldn't be captured and they shouldn't be retained by the baits. In blue, we have the target that's retained during enrichment. And here we have a target that contains the PCR sequence for which we're going to be quantifying before and after hybridization. So essentially by quantifying the total library before and after hybridization, as well as this target associated sequence, we can generate a number to get an idea for the fold enrichment of this specific PCR sequence, PCRable sequence, relative to the total library. In an ideal world, we want to see a massive depletion of the background library and a comparatively much smaller depletion of the target sequence here. And in reality, that's actually exactly what we see. So based on relative quantification through PCR with a 1 hour hybridization at different hybridization temperatures in the presence of 55% ethanol, we see an increasing fold enrichment with hybridization temperature. So much so that at 45 degrees, which is the highest temperature we tested, we have over 1,000-fold enrichment in the on target or in the target molecule relative to the background. So this is really exciting, but again, it's only preliminary work. We won't know for sure the effectiveness of this protocol until we have sequencing data, which is going on the machine now. Clearly, hybridization temperature has a big impact on efficiency, and I've confirmed ethanol's impact during previous testing. So I did this test with another bait set and another sample, looking and seeing that after 1 hour, there was no significant enrichment with 0 ethanol, and there was, I think, 80-fold enrichment with 55% ethanol. So that's what we made us choose this for this test here. But again, these results are very promising, and we're really excited to continue optimizing and testing this reaction, hopefully implementing it in our workflow down the road. So in conclusion, our proposed workflow circumvents many of the problems associated with DNA sequencing. We've shown that baits made with hub design work reliably and detected low levels of pathogen DNA against a human background. And we're currently optimizing a 1 hour hybridization step to allow for enrichment without an onerous time commitment. We anticipate that when combined, our workflow will allow for the robust detection of causative organisms from the blood of patients with sepsis, as well as AMR profiling in less than 8 hours. For acknowledgements, I just want to thank my committee, Dr. Henry Poinar, Dr. Jerry Wright, and Dr. Jacob Megalin, as well as my colleagues, Zachary Dixon and Melly Cook. They have been absolutely instrumental in the bioinformatic and wet lab processing of these samples. Thanks for having me. That's all I have for today.
Thank you, Dirk. That was great. Really exciting. So while we wait for some questions to come in, I have a few for you guys. I guess I'll start with Dirk since you were most recently talking. So Jeff does mention that cost was an issue. So what is the cost of doing your assay relative to the standard?
Expensive, incredibly expensive, more expensive than the thing that Dr. Pernica said was too expensive. So, and this is not the final version of this protocol. Right now, the sequencing technology isn't at a place where it can be implemented on the level that it needs to be. However, we're confident by eventually adapting this to technologies like nanopore and other sequencing technologies that are emerging. I know Illumina is supposed to be coming out with a rapid sequencing technology fairly soon, which should hopefully have a lower price. But we're confident that this will be able to be implemented down the road with the implementation of those technologies.
And of the steps that are currently done in your lab, are any of those technologies already in a clinical lab so that, you know, the adoption of this new technology would be less onerous?
So putting this into a clinical lab will also require automation steps. So you need to have a machine that will be able to, you give it a sample and then it processes the sample all the way through, because it's a complicated workflow to do for somebody to do on their own. I don't believe sequencing, other than potentially Sanger sequencing for some molecular tests, has been implemented on a large scale. Though I might be wrong on that. I'm sure some people know more. But again, this is, I believe, where the technology will eventually lead and where molecular testing will get to in the next 10 or so years.
And,
at least I hope
so.
Yeah.
Is there a way to couple the bait capture to another type of detection technology that is not sequencing? So, I was thinking, like, I realize you're already using hybridization, but could you, I was thinking more like fish or some kind of a chip where the hybridization itself is the diagnostic as opposed to just enriching and then sequencing.
So we actually have an industry partner that we're hoping to collaborate with. And so we're very early stages on that. So I didn't know how much I'm actually allowed to talk about it, though I am allowed to say,
kill us.
Yeah. Though I am allowed to say we do have a potential industry partner, and we're looking at exactly that, being able to use the hybridization, as well as we're looking into some other molecular diagnostics for far more rapid detection, though maybe not as sensitive detection. And that's things like Cas12 detector, Sherlock, these sorts of newly developed assays.
Yeah, it's a very cool time, and I think the COVID pandemic has also really pushed the diagnostic field a lot faster than it might otherwise have.
It's a very cool time to be working in diagnostics.
Yeah, no kidding. So I have a question for Des as well, related more to I guess your comments on, even if you give people information, you can't make them use it. You can lead a horse to water, but you can't make them drink. So, I realize that this is a problem in Botswana, but do you also find this problem even in better resource countries like Canada, where, people who are used to doing things a certain way are resistant to change when it comes to, you can give them the BioFire results here as well, and they may not use it because they have a pattern in their head of how things should go and what they're used to doing.
Absolutely. People are used to doing what they're doing. That is, as you say, right? Like, that is not restricted to Botswana. Like, I think the take home from my thing should not be that, like, Botswana is different, but that the context is really critical. And if you want to think about a Canadian a Canadian example of how context differs, like, the BioFire would be insane to do in Hamilton. Well, maybe not insane. Let me rephrase. I am not sure how much incremental benefit there would be to doing the BioFire off positive blood cultures in the middle of Hamilton, right? For the reasons that we have all these infectious disease, we have all this tech already. It would be much different in a Iqaluit or somewhere where, somewhere remote, where you have sick people, but you don't have a local lab. Like, the last time I checked, they were flying all their stuff to Edmonton, right? But I think I complain about the length of time it takes for samples to get from Mumpsy to the general, because they have to wait for the truck. But that's a whole, like, that's multiple orders of magnitude different. And so if the alternative is people doing worse and then having to be medevaced to Montreal, what is too expensive is very, very much different. And I think you have a great point that humans are going to be humans every place, and the benefit of diagnostics is going to be dependent on exactly what the clinical scenario is, because it's not just sepsis, right? Like, there's all sorts of different kinds of infectious disease scenarios where I think diagnostics offers the potential to improve care, but we always have to work on people.
Well, and I know from talking to you in other scenarios that people are treating syndromes, right? So they have a pattern that they follow because it's successful. So I guess this comes down to education, right?
Yeah, the education that they initially got, the way that it is now, like, I feel like I'm not so old, but even when I trained, like it was 20 years ago, right? And stuff's a lot different now. So stuff that might have been appropriate then may very well not be appropriate now. And it's hard for people to get used to that sort of thing.
Mm-hmm. Okay, so we have a couple of a couple of questions. Sorry, I need more coffee. The first one is from Kate Kennedy. She says, hi, Dirk, great talk. Since there are microbial contaminants in your sepsis blanks, will the ethanol hybridized bait capture potentially also make it harder to tell a blank from a low CFU positive? Versus mastodon, where you're unlikely to have contaminants that match your baits?
So yes, but again, looking at the blank that we had in that one slide relative to the low spike sample, there's still around a 10-fold difference even in the enriched fraction for the blank relative to the enriched fraction for the low spike sample. And so it's impossible to, so I think we got around 2 million reads on that run specifically for each of the samples. And those, the proportions of those different reads for the blank were, I believe, on the order of 1 to the -5. So that's around 10 reads out of 1 million. So it's an incredibly low proportion. And this is where it starts to get very difficult, especially since these were processed in parallel with all these, the high spike sample, which is 1 million. It's very possible that some DNA can just float over from one tube to another. So this is about figuring out a way that we can look at maybe not even just the proportion and the amount, but we can actually look at the different damage signals, the different size distribution, other things that are associated with this to better distinguish blanks from, or false positives from true positives.
Thank you. And there's a second question from Kathy. She says, did you try higher percentages of alcohol before proceeding with 55%?
We didn't try higher percentages of alcohol because the issue is that at a certain point, it starts to precipitate the DNA. So it gets to, like a peak of efficiency, and then it goes, because then you get no DNA carryover at all. And this is something that they showed in the original paper. So with further testing and with preliminary, and when we go beyond the preliminary work that we've done so far, I do plan to really fine tune and say, okay, we'll do 55, 57.5, 60, and just see what that peak exactly is. But for right now, I just wanted to disseminate some of the preliminary work and show that we have some promising stuff, some promising initial results.
And I have to say, I wasn't shocked to see that there were bacterial reads in your blank blood sample. I mean, I think it would be very difficult, especially if you're using a really sensitive technique, to collect blood without any bacterial contamination, right?
Well, especially, I mean, I remember Dr. Pernica's presentation, I think it was 60% of the blood cultures were contaminated.
Yeah, that I don't think would be the standard around here. For
yes, for sure. For sure, but
there'd be questions.
But if you were to implement this in a setting such as that, when it gets cheaper, then I think this sort of thing where you can look at the different damage signals, et cetera, and all the other information you get with DNA sequencing, you have a much better chance of distinguishing a true positive from a false positive.
Well, how do you decide what's a contaminant? Like, is it just based on numbers?
For DNA sequencing, the original paper that we were looking at was looking at an SIQ, which I believe is a standard identification quantifier. Anyways, it was a number that they generated to distinguish false positives from true positives. And it worked fairly well, although that's an area of active research that we're working on as well to try to figure out what's the best way to truly distinguish those two groups.
Okay, Dr. Fulford says we still get a surprising number of contaminants here. Don't know if you want to admit that in a public forum like this. Don Bowdish said, great talks, both of you. Best of luck with this work. It is so important. We need innovation in diagnosis of respiratory infections too. And Padman said, even without contamination, there's extraneous DNA in normal blood that would be picked up by sequencing. And I totally agree. I mean, you can't, especially if it's collected in the normal way. I don't know how you get by having some bacteria introduced into your sample. Okay, I'm not seeing any further questions, and we are at the end of the hour. So thank you so much, both of you, for those really interesting talks. And we will see everybody in March.